Have you ever thought about sleep? Maybe not, because sleep is a naturally recurring state of mind and body. However, have you ever wondered why we need to sleep or what is happening to our bodies during sleep? In fact, nobody has a clear answer to these questions. Sleep is still full of mysteries. In the Lazarus Oishi Lab, we are working on solving the mystery of sleep. We focus in particular on the following topics: molecular and cellular links between sleep and motivation, discovery of drugs to treat insomnia and schizophrenia, and elucidating sleep function and regulation by using short sleeping mice. Let's take a closer look at these three topics. We can all lose ourselves in our favourite things without feeling sleepy at all. However, the brain mechanisms governing the regulation of sleep by cognitive and emotional factors, such as excitement or boredom, are not yet well understood. The nucleus accumbens, or simply the accumbens, part of the brain, is associated with motivation and pleasure. Since the accumbens has many neurons that express receptors for adenosine, a brain substance that induces sleep, we thought it might play a role in linking motivation and sleep-wake regulation. We use chemogenetic and optical techniques to remotely control the activities of adenosine A2A receptor expressing neurons in the accumbens. When we activated these neurons, sleep was strongly induced. We also conducted other experiments. We tried to motivate mice by giving them their favourite chocolate or toys, or putting the male mice together with female mice. We observed that the activity of these neurons and the amount of sleep decreased. These findings indicate that the activity of adenosine A2A receptor expressing neurons in the accumbens is important for sleep-wake control and is linked to motivation. Speaking of sleepiness, many people drink coffee to wake up in the morning or to counteract fatigue during the day. Coffee and tea contain caffeine, which is the most consumed psychostimulant in the world. Although its arousal effect is well known, the mechanism as to how caffeine wakes us up has long been a mystery. We have shown that caffeine produces its arousal effect because it prevents adenosine from binding to adenosine A2A receptors in the accumbens. It is said that 10 to 15% of the world's population and 30 to 60% of the elderly are suffering from insomnia. Drugs are used to treat insomnia, but these have many adverse effects and can lead to addiction. So, there is a need for drugs that promote natural sleep without harmful side effects. Although agonists that activate A2A receptors like adenosine strongly induce sleep, adverse cardiovascular effects preclude their use in treating sleep disorders. Therefore, as a new approach, we focused on positive allosteric modulation. In this approach, a drug and adenosine bind at different sites of the adenosine A2A receptor, resulting in enhanced receptor activity. We identified a novel compound that increases sleep by enhancing the action of endogenous adenosine. We demonstrated that enhancing A2A receptor signaling with this compound induces sleep. In addition, unlike A2A receptor agonists, our compound has no cardiovascular side effects. Allosteric modulators for the A2A receptor may help people with sleep problems to fall asleep. We spend about a third of our lives sleeping. Recently, it has been found that the lack of sleep not only reduces concentration and performance, but also increases the risk of high blood pressure and dementia. Although we all know sleep is important, research on understanding the need for sleep has not been successful. One of the reasons is the lack of suitable model organisms. Recently, we accidentally created mice that require an extremely low amount of sleep. Surprisingly, these mice do not show an increase in a specific marker for sleepiness. Therefore, the behavior is very similar to that of so-called short sleepers, people who can function for long periods of time on little sleep without exhibiting excessive sleepiness. Using such short sleep models, we try to understand the necessity of sleep. We also study the neural mechanisms of short sleep to clarify the control mechanisms of sleep. Since we are targeting new and unexplored mechanisms, our studies may lead us to the development of new sleeping drugs. In our lab, a team of scientists, technical staff and students are conducting highly collaborative research by using cutting-edge technologies, for example, genetically engineered systems and in vivo imaging. English is the official language in the lab. English speaking and reading abilities are essential for researchers in neuroscience.
We welcome students with great curiosity in neuroscience and motivation to learn cutting-edge technologies, and of course the desire to do research in an international environment. Students who look for opportunities to collaborate with medicinal chemists on the discovery of new drugs for sleep or psychiatric disorders are also welcome.
So if you are interested in why we need to sleep or how we sleep, please join our laboratory.